Hi, I'm Jessica Bowser. I'm a tenure track assistant professor at the University of North Carolina School of Medicine in Chapel Hill, North Carolina, and I'm a 2017 IARS Mentor Research Award recipient. I'm incredibly grateful to the Society for their support and the individuals that make the IARS Mentor Research Award possible. I grew up in the Midwest. I'm a farm girl. In the Midwest, our school was actually an old Atlas missile silo. It is the only Atlas missile silo in the world that is used as a school. Living on the Midwest is hard work, dedication, commitment, working as a team. I think I've learned in the last several years, not only from my time in science, but I think even my time in the Midwest, that you can do more as a group and as a team. And I think that team commitment makes all the difference in the world.
In 2017, as an instructor at the University of Texas McGovern Medical School, Dr. Jessica Bowser was awarded the IARS Mentored Research Award for her research targeting epithelial regeneration for perioperative organ protection.
What drew me to this area of research was the idea that I was training in the background of cancer biology at that time. I was discovering new biology that was important to the pathogenesis of that type of disease. But at the same time, as I was coming on new knowledge that was important for cancer, I was finding that my research was actually providing important insight to understand how normal epithelial cells regenerate in conditions of tissue damage and acute and chronic inflammation. And I wanted to apply these important findings for innovative solutions to work in an area of medicine that faces the challenge of dealing with chronic or acute inflammation that leads to devastating patient outcomes. So the initial goal was really to identify molecular targets or pathways that are essential for promoting regeneration of epithelial cells on tissues that are damaged by acute and chronic inflammation. But I was trying to find a target that could be directly targeted by drugs, with the rationale of trying to identify druggable targets to promote epithelial regeneration that we could exploit to prevent and overcome devastating inflammation that leads to life-threatening conditions such as sepsis, multi-organ dysfunction, or ARDS. And all these conditions are primarily due to the breakdown of epithelial barriers. And currently, there are no approaches that directly promote epithelial regeneration, especially approaches that could be used in critical care medicine or in a perioperative setting. We were able to use sophisticated models in the lab in which we can induce tissue inflammation in the gut to break down epithelial barriers and to have those epithelial cells regenerate. And during that process, I was able to discover a novel molecular target and a corresponding signaling pathway that are essential for epithelial regeneration. And not that they could just be important for epithelial regeneration, but again, that they were able to be targeted by drugs. And not drugs that we just use as compounds in our lab, but actually by pharmaceutical agents that are widely available and currently in use in clinical trials for other types of situations in different diseases or human health conditions. We're closer to understanding and developing druggable approaches that will directly promote epithelial regeneration, which potentially could be used in the real-time clinical settings, such as perioperative medicine or clinical care medicine, because current regenerative approaches are not optimal for these areas of critical care or emergency medicine. So hopefully with druggable approaches, we can actually now expand to more real-time clinical settings. The science has evolved. We are now working in new areas such as genomics and metabolomics. Really, this has allowed us to ask questions that are essential to understanding the clinical benefits and potential shortcomings of using drugs to target epithelial regeneration. We're incredibly excited about the next steps of this work, but again, this would not be possible without having the support of the society for building this research and making opportunities possible to connect with collaborators to push this work forward. It opened doors for me professionally. It provided an upward movement in my career that would have not been possible without the support of that IARS Mentor Research Award. I was able to secure a competitive tenure track appointment here at the University of North Carolina School of Medicine with a very generous startup package that allowed me to fully develop a research program that stems from that initial work I did with the Mentor Research Award. It also allowed me to develop very competitive R01 NIH applications, which one recently was favorably scored. But it additionally allowed me to establish national and international collaborations that have really pushed the work forward and helped me to also form essential relationships that now I'm part of other investigators' competitive grant applications to push not only work related to my work forward, but even work beyond that. I know that there are patients out there or loved ones of patients that are hoping for solutions, that will help them immediately. So it's really hard just to wait to watch the science develop and to evolve. I think that's the challenge. But I think also what is the rewarding side of this is the flip side. I think it's seeing the advancements of the science that I know one day with not only myself but others who work in this same area will make significant impacts for patients and will be something that I think will really benefit not only the area of anesthesiology, but I think medicine in general.
Your IARS membership and donations enable the support of future research leaders who will advance the science. Thank you.